Hello students, it's me G Trupti. As we know, there is only 1 month left over for the final NEET exam. I know definitely most of you students are in worry regarding how to recall every lesson of zoology. So definitely don't worry. From my side, I'm planning for all my students to give a small crash course. You could call it as Recall Zoology with me. Okay, so all the best. Feel always that you are the best and do the recalling. Okay, you have done till now number of lessons and all, but there is need always to recall each and every lesson very perfectly. So keeping this particular thing in my mind, I'm going to start for you one special course, that is Recall Zoology with me. So let's start with the first topic. The first topic which I'm going to start today for you only is the animal kingdom. Okay, kingdom Animalia. So the weightage for this lesson, if you say minimum 2 questions or maximum 4 questions are seen till now. So based on that you could understand the weightage. Minimum 2 questions, maximum 4 questions. Let me start then. Whatever this lesson is there, this particular kingdom Animalia is getting divided into 3 different parts, you could say. The first one includes basics. The second one includes animal diversity 1 or invertebrates phyla right from Porifera till Hemichordata, and the last one it includes the phylum Chordata right from Protochordates till the class Mammalia. Okay, so let me start first of all the basics today. Right, so here kingdom Animalia. First of all, what we have to check here, mainly I will tell you, because this is a recalling course. Here there nothing will be there in the detail, expecting that you people have done it very well previously. So in the kingdom Animalia, the basic things which you should do, which you should know, first of all the terminologies, like what is the meaning of, based on the primary germ layers, the Diploblastic group, Diploblastica, Triploblastica. Terminologies like this are damn necessary. Symmetry, level of organization, coelom, these are the main things, first of all, which we are going to cover here in short. Okay, so let me start then. Let me start here. Kingdom Animalia is also known as metazoa. As you know, to call one living entity as animal, that living entity should fulfill 3 different criteria. Those criteria are, I could write here, a living entity which is multicellular, which is eukaryotic, and which is heterotrophic in nature. Heterotrophic in nature, you could call that living entity as animal. Yes, so the living entities who are fulfilling these 3 major criteria, they are coming in the kingdom Animalia. You could call them as metazoa also. Okay, all the animals are again not same. So again the kingdom Animalia is getting divided into 2 subkingdoms, and as you know, this kingdom Animalia is getting divided into 2 subkingdoms. They are, the first one, that is Parazoa, subkingdom Parazoa, and another, that is subkingdom Eumetazoa. Eu means true animals. Eumetazoa. You know, Parazoa include the only one phylum, that is phylum Porifera, which includes examples like sponges. You know, whatever parazoans are there, whatever poriferans are there, as they are fulfilling all these criteria, we have took them in the kingdom Animalia. But again, why we have kept them apart from leftover all animals? Because they have only cells in their body. There is no any tissue formation seen. So they are kept in a different subdivision, subkingdom. You could call it as subkingdom parazoa, right? Concentrate here on the eumetazoa. What are eumetazoans are there? Definitely they are true animals. Why we are calling them as true animals? Because they are having tissue formations in their body. Tissue formation means primary germ layers in their body. Again, how many primary germ layers? Based on that, eumetazoa is getting divided into two groups. So what your subkingdom eumetazoa is there? It is getting divided into two grades based on how many primary germ layers are there. Definitely one, that is Diploblastica, having two primary germ layers as you know, and the Triploblastica, grade Triploblastica, having three primary germ layers in their body. So here Diploblastica are having ectoderm and the endoderm, two primary germ layers, while Triploblastica will be having ectoderm, endoderm, and in between them you will be getting the mesoderm. So endoderm, then this is the ectoderm, and in between these two layers you are getting one more layer, that is mesoderm, primary germ layers. So who are having two primary germ layers, they are coming under the grade Diploblastica, and who are having three primary germ layers, they are also known as Triploblastica. This is one criteria for the classification as you know, and another criteria for the classification of these animals is their symmetry. So as you know, symmetry is nothing but the arrangement of paired body parts along the axis of the body. So symmetry terminology, you know, simple way I could say, those animals we are able to cut them in exactly two equal parts. Two equal parts are known as antimirrors of each other. All those animals to whom we could cut and we are able to get exactly two equal halves, antimirrors of their body, you could call those animals as symmetrical. So we know in the kingdom Animalia, except Porifera, leftover all the animals are having their symmetry. All the animals are symmetrical with a few exceptional cases. But again, whatever that symmetry is there, it is of two types as you know. One, that is bilateral symmetry, other that is radial symmetry. Bilateral symmetry is one which is the main symmetry of all the triploblastic animals. So remember one thing, all the triploblastic animals are having bilateral symmetry with a few exceptional cases in them. Triploblastic animals who are having three primary germ layers, you could say they have bilateral symmetry. Means what? Bilateral symmetry is one in which only one way is there to get exactly two equal halves of the body. Only one way is there, and that one way is median sagittal plane. For our body also same thing. If you want to get exactly equal two parts of our body from this way only, this way, you could call it as median sagittal plane. Through that plane only we should cut our body. Then we are able to get right and left equal halves of the body known as antimirrors. So you could say all the triploblastic animals are having bilateral symmetry. So bilateria, you could call this grade as bilateria also. And the diploblastic animals, they are having some other type of symmetry. You could call it as radial symmetry. So we could call them as radiata also. What is the radial symmetry? As you know, when more than one ways are there to get exactly equal two halves of the body, more than one ways, then you could call that symmetry as radial symmetry. So you could say radial symmetry is the principal type of symmetry present for all the diploblastic animals, and the bilateral symmetry is the principal type of symmetry present in all the triploblastic animals. You could remember one short form here, okay, like in a hurry you should not make a mistake. So I could tell you to understand in this way. You could remember it in this way: doctor of TB. Doctor of TB means what? Doctor of TB in the sense all the diploblastic animals have radial type of symmetry, while all the triploblastic animals will have bilateral symmetry. with some exceptional cases in them. Okay, so in the Diploblastic group, Radiata, you will be getting only two phyla. They are phylum Cnidaria, which includes the major examples like Hydra, jellyfish, phylum Ctenophora includes main example sea gooseberries. Okay, so these animals are having radial symmetry, you could say. In that, one more thing I could add up, whatever your sea anemone, if I write sea anemone is also coming under phylum Cnidaria. So sea anemone and the sea gooseberries, or you could say ctenophores, will be having biradial type of symmetry. And what is that biradial symmetry? It is one type of radial symmetry only in which two ways are there to get exactly equal two parts of the body. So simply remember, all the diploblastic animals are having radial type of symmetry, and all the triploblastic animals are having bilateral symmetry with few exceptional cases. So let me talk about what are the triploblastic phyla. So the first triploblastic phylum here you are getting, that is Platyhelminthes, which include flatworms. Then Nematoda or Ascahelminthes, which include roundworms. Then Annelida, which include earthworm. Arthropoda, all the insects are coming here as a simple example. Mollusca, octopus will be coming here as an example. Echinodermata, starfishes. Hemichordata and chordata. And chordata include protochordates and param. What is this param? Okay. P stands for Pisces, fishes, amphibians, reptiles, aves and the mammals. So the first triploblastic phylum or the first bilaterally symmetrical phylum, you could say it is Platyhelminthes, and it is ending somewhere in the phylum Chordata. Few exceptional cases, you will be getting the exceptional cases. I am putting star mark here for the molluscans and the echinoderms. You could say one statement for the echinoderms. For the echinoderms, remember the main example are the starfishes. For the echinoderms, I could say one statement. All the echinoderms are primarily bilateral. All the echinoderms are primarily bilateral, but secondarily became pentamerous radial. Means what? All the echinoderms in their larval stage have the bilateral symmetry, but as they became adult, they lost their original symmetry and they became pentamerous radial. So all the echinoderms are exceptional case, you could say. So in the exam also, based on either they are asking the larval stage or adult, you have to decide its a symmetry. But mainly you could say in the larval stage they are bilaterally symmetrical only. So if they are mentioning adult, then you have to write the pentamerous radial symmetry. In the molluscans, only one example is there, that is gastropodas. Gastropoda. So gastropoda include snail. Gastropoda include main example, what is that? Snail. So for the gastropodas, not for the all molluscans, please remember, only for the gastropodas where snail is the main example. For gastropodas, statement I will use is all the gastropodas are primarily bilateral but secondarily became asymmetrical, means they lost their symmetry as they metamorphosed into adult. It means in their larval stage they had the bilateral symmetry, and as they became adult after metamorphosis they lost their symmetry. So after becoming adult, in any way you try to cut them, you will never get exactly equal two parts of their body, right? So here you could remember with respect to the symmetry, poriferans, what are poriferans are there? Here with respect to symmetry I will write most of them are asymmetrical. most of them are asymmetrical, means some of them are having a little radial type of symmetry, so they have given it like that. So in the parazoa, with respect to symmetry and all, if you talk here with level of what level of organizations and all are there, here you could say in the parazoa, phylum Porifera is coming. They have only cells in their body, they don't have any tissue, so that we kept them aside. But here, right from eumetazoa, means what are real animals are there, we call them as true animals. They have tissue layers in their body, but based on how many tissues, thing is 2 layers, Diploblastica, 3 layers, Triploblastica. And based on symmetry, radial symmetry, bilateral symmetry. 2 phyla here, Cnidaria and Ctenophora, with the examples which I have marked here. And here also all the triploblastic animals are having bilateral symmetry with few exceptional cases. So here Doctor of TB is the formula which you could remember for your easy understanding. I hope till here you understood. We have to continue with the classification.
Ready.
So we will check here the further classification of kingdom Animalia right from the Triploblastica grade, or based on the symmetry it is known as bilateria. So what are triploblastic all phyla here, which we have written here on this side. So whatever these all phyla are there, though all of them are having 3 primary germ layers and having the same type of symmetry, again they are getting divided into 2 different groups based on the fate of the blastopore. So based on fate of blastopore, which is the opening of Archenteron, as you know. So first group is Protostomia and other group is Deuterostomia. Protostomes, proto means first, stoma means mouth. So animals in which blastopore develops into mouth, deuterostomes, deutero means secondary, stoma means mouth, blastopore developed into anus. So here, what are protostomes are there? Animals in which blastopore is developing into mouth. Okay. So what are the examples here? So in the protostomes, you will be getting the phylum like Platyhelminthes, Nematoda, Annelida, Arthropoda, Mollusca. What are these 5 phyla are there? They are coming under the group Protostomia. Okay, they are coming under the group what? Protostomia. So here I will write Platyhelminthes, Nematoda, Annelida, Arthropoda, Mollusca. All these are the phyla who are coming under Protostomia group. Though they are protostomes, again they are different from each other. So they are further getting divided into again another categories based on the body cavity. So what is that? We will come to that point next. Deuterostomes are the animals in which the blastopore developed into anus. So here you will be getting the examples: Echinodermata, Hemichordata and the Chordata. These are the 3 phyla. So further again they are getting divided into different divisions, different groups based on the body cavity. And body cavity is nothing but coelom, you know. So the animals who are having 3 layers, we expect in their body presence of a cavity known as coelom. So simply I could tell you what a coelom is there. It is a body cavity which is a space bordered by 2 mesodermal layers. This particular cavity is doing number of functions. It is mainly helping to give protection to the internal organs, giving little space for the movement of internal organs. More than that, many functions are there, as you know. So here, whatever based on that body cavity or coelom, again there is a further division is there of this particular protostomes. So the first group you will be getting, that is acoelomate. Acoelomata or acoelomate. For example, phylum Platyhelminthes. Second, you will be getting pseudocoelom. Pseudo means false body cavity. Pseudocoelomate, animals with false body cavity. Acoelomate, there is no body cavity. Here you could say they have ectoderm, endoderm. In between here, you will be getting a solid mass. So no cavity. Pseudocoelom, ectoderm is there, endoderm is there, and one muscle layer you are getting a false body cavity is there, and then you will be getting a type of true coelom, that is cygocoelom. Cygocoelom is one which is formed by splitting of mesodermal block. Splitting of mesodermal block. The coelom formed by splitting of mesodermal block is known as cygocoelom. So here Annelida, Arthropoda and Mollusca, these are the three examples. Ectoderm is there, endoderm is there. This is the outer mesoderm, as you know, and this will be a inner mesoderm. And in between these two mesodermal layers, whatever space is there, that is a true coelom. So you could say this is a true coelom. First eucoelomate is Annelida, or first cygocoelomate is also Annelida. Now come to these particular deuterostomes. You could say in case of deuterostomes, you will be getting only one type of coelom, that is enterocoelom. Enterocoelom, which is formed by outpouching of the mesodermal blocks. Outpouching of the outpouching from the enteric region. Outpouching happened from the archenteron. So that is the enterocoelom, and the examples are same: echinodermata, hemichordata and the chordata. So here few statements I could say, please remember these statements. I could say all the enterocoelomates are deuterostomes, or I could say all the deuterostomes are enterocoelomates. It will be one and same. Other thing which you should remember perfectly are, first true coelomate phyla is Annelida, or you could say first cygocoelomate phyla is Annelida. Here I could say all the protostomes are not eucoelomate. Protostomes include acoelomate, pseudocoelomate and cygocoelomate. If I say the statement all the cygocoelomates are protostomes, it will be a definitely correct statement, but I can't say all the protostomes are only cygocoelomate. In the same way, I could say all the pseudocoelomates are protostomes, all the acoelomates are protostomes, but I can't say all the protostomes are only either acoelomate or pseudocoelomate or cygocoelomate, because protostomia group contains three different types of organisms based on the body cavity. Some are acoelomate, some are pseudocoelomate and some are the cygocoelomate. But here the statement will be true. All the enterocoelomates are deuterostomes, or the deuterostomes are enterocoelomates. So this is regarding the basics, and with one last point I will finish this with level of organization. So levels of organization, that is one more point to remember here. So level of organization means what? Very simply we could say, right from porifera till the phylum chordata, in all the animals, definitely they are doing all the physiological functions. But who is doing that physiological function? Who is carrying that particular function? Based on that, four levels of organizations are there. Either cell is carrying that function, or tissue, organ is carrying, or organ system is carrying that function. Based on that, four levels of organizations are there. So I could say here cellular level of organization, cellular level of organization, tissue level of organization. So here we will take one more point in the basic, that is levels of organization. As I told you, based on who is carrying the physiological function, we are having four different levels of organization with their examples. Cellular level of organization is one in which cells are doing the different types of works. Distribution of labor is there. So the best example, the one example which we have for that is phylum Porifera, includes sponges. Tissue level of organization is one which is you could say more advanced as compared to the cellular level of organization, or lowest level of organization in Eumetazoa. Eumetazoa, lowest level of organization in Eumetazoa is tissue level of organization, where tissues are doing the special type of functions. So here you will be getting the two phyla, Cnidaria and the Ctenophora. The third one, organ level of organization. It is quite advanced as compared to the tissue level, in which tissue is united to form the organ, and the organs are doing different sort of functions. So the phylum Platyhelminthes is coming under that. And the last one, that is highest level of organization in kingdom Animalia, that is organ system level of organization. It is there right from the nematodes, or you should call them as Ascahelminthes, till the phylum Chordata. All of them, they are having number of organs which are like working together in a coordinated fashion to carry one physiological function. So highest level of organization in kingdom Animalia will be the organ system level of organization, and the lowest level of organization in kingdom Animalia will be cellular level of organization. But if specifically they ask the lowest level in Eumetazoa, then you should write it is a tissue level of organization. One more level of organization I could say, that is protoplasmic level of organization, which is there only for the protozoans, not for the animals. Protozoans like amoeba, paramecium, euglena, which is single celled entities, unicellular, whatever entities are there, for them you will be getting the level of organization, that is protoplasmic level of organization.